This is the calculation video for the Newcombe lab. In this video, we'll go over how to work out the problems in the Newcombe lab and how to figure out your mutation frequency. A lot of this is uh, presented in the lab manual, and so you may not need this video, but if you're having any trouble with it, this will help get you through it. So the um, big question that the big calculation, I should say, that the Newcombe experiment asks you to do is to calculate the mutation frequency. The big thought questions are, it asks you about the cause of the mutation, is it spontaneous or induced, and it asks you about, can you tell what type of mutation, and those are a little more, esoteric, right? A little more, what's going on. But the calculation is mutation frequency. And so how do we approach the mutation frequency? Well, what we're going to need is two numbers. We're going to need to know how many mutants there are, and we're going to need to know the total number of cells. Okay? That's going to be the mutant concentration and the viable cell count. We're going to deal with concentrations here, so mutant concentration instead of total number of mutants, and the cell concentration, total cell concentration, instead of the total number of cells, because we're dealing with bacteria and they're very small and it's very hard to get absolute numbers, and it's a lot easier to deal with concentrations than it is absolute numbers. But other than that, this is a very simple calculation, right? If you wanted to know what the frequency of red M&Ms in a bag or a jar of M&Ms was, you'd count up the number of red M&Ms and divide it by the total of all of the M&Ms in the bag or the jar, and that would be the frequency of red M&Ms. And so that's what we're doing here. We're going to count up the mutants and divide it by the total, but we're going to use concentrations. Concentration of mutants and the concentration of all. And so that's it. And so how do we get these numbers? Well, that's what you used. That's what you did the experiment for. We're going to use those plates for. Okay, so to get the total number of cells or total concentration of cells, the viable cell count, what we did is we took E. coli B and we plated it. Now the problem is it's a very high concentration, and so if we just plated it as it was originally given to you, what you'd get is a lawn of cells. It would just completely fill up the plate. You'd get a confluent growth. It would all grow together into one big giant lawn is what we call it, but one big giant, it would fill it up, right? One big giant colony. So that's a problem. We need something that's countable. So what you did was you diluted it first by a known amount so that you could get a countable number of colonies, and then you could work backwards, right? You could say, all right, well, we know we diluted it by this much. That gave us so many colonies. And then you can multiply backwards to say, well, what was it in the original sample? And that will give you the viable cell count. For the mutant concentration, what you did was you put a selective pressure on those E. coli B cells. You did that by taking the E. coli B and without diluting it, because a lot of them are going to die. In fact, the vast majority of them are going to die. You plated them on plates that you then added the bacteriophage T2 to. And the T2 was able to kill most of the E. coli B, but not quite all of them. And the ones that survived were the mutants. And what we have to do is just count them up. And when we count them up, we can see how often it happened by dividing the bottom number here by the top number. So the mutant concentration is going to be the easier of the two to figure out, right? Because there's no real big calculations there, no dilutions. And so what you did was you took your E. coli, you plated it on two different plates because you actually had a treatment going on. Um, you put 100 microliters of the E. coli B on two different plates. You let it sit there for 2 hours, and then you respread one of those plates, and then you added T2 to both of those plates. You let it sit at least overnight. You looked at it late the next day, maybe 2 days, and you counted colonies. And for some of you that was really easy. For some of you, you had really small colonies and really large colonies, and you had to kind of set a cutoff. But whatever it is, you got your colony count. And let's say, for example, what you found was 50 colonies on the not respread, the one that you left alone, and 120 colonies on the respread plate. Now that may not be what you found. Another possibility is that you found roughly the same number of colonies on both plates. Maybe it was 100 and 100. That's another possibility. Remember, the result tells you something about whether the mutation is induced by the selective agent, in this case T2, or is is present before the selective agent is added to the bacteria, which tells you that the mutation was spontaneous, that it happened before even seeing the selective agent. We'll go through that in just a moment, but let's just presume here that this is what you saw, 50 and 120. If they're the same, then it doesn't matter which plate you use because they're the same. But if they're different, then we have a question, don't we? Which plate do we use? There are a lot of possibilities, aren't there? I mean, do we use the not respread plate, 50? Is that the number we should use for our calculations? Do we use the respread plate, 120? Do we average them? Is it 170, 50 plus 20 divided by 2? Is that the answer? Is it 170 total mutants that we should use? That's what was on both plates. So how do we proceed here? Well, the answer is we want to use the plate that represents the actual number of mutants in the original tube when we did the experiment, right? We took sample from here, 100 microliters of it, and put it on these two plates, and then asked how many mutants we had. Well, if we had just done that and left it alone, how many mutants would we have had? Well, the answer is 50, right? Because to get 120, we had to manipulate it further. We had to allow them to grow into nascent colonies, into little baby colonies, micro colonies, and then break them up and spread them around the plate so that they could form new colonies. But they wouldn't have been able to do that unless we reseeded them all over the plate. So there weren't really 120 mutants in that initial sample, were there? There were 50 mutants. And so 120 is inaccurate. So is the average between 50 and 120, and so is 170 per 200 microliters, which is just the average, or 170 per 100 microliters. All of those are wrong. The real answer for both of these plates is that about 50 mutants per 100 microliters is what was put on the plate. And so that's what we're going to want to use. 50 mutants per 100 microliters is what we're going to use for our count. Okay, and so we'll move that forward to when we do our calculation. And so how do we figure out whether this tells us whether it's induced or spontaneous before we move to the viable cell count? Well, remember there are two possibilities. It is not going to be both of these things. It's going to be one or the other. Okay? The mutation is either caused by the selective agent, it's induced, or it is spontaneous and it has nothing to do with the selective agent being present. And so the only reason we're drawing both of these things out is because they give you different results. And so what you're going to do is you're going to see one of these results or the other result. And so let's see what these two possibilities give us. Now, a real mutation frequency, you know, is something like 1 in a million, or 1 in 100,000, or 1 in 100 million, you know, depending on the type of mutation. That would be a lot of cells to draw on the plate. We are dealing with that many, right? I mean, you have a viable cell count probably of somewhere in 10 to the 8th or 10 to the 9th, and then you're putting 100 microliters on these plates. So you are dealing with millions and millions of cells, but we're not going to draw that many. We're only going to draw a handful of cells on each of these plates. So let's pretend that our mutation frequency is much, much, much higher, something like 1 in 4, and we'll just go with that mutation frequency for both conditions, induced or spontaneous, and see, you know, what happens if we start with 4 cells on both plates. And then what happens if if the mutation is induced or if it's spontaneous. So let's just start by putting cells on all the plates. Whether it's induced or spontaneous, we'll put 4 cells on each plate. So these are E. coli B cells. And whether it's induced or spontaneous, if you then incubate them for a couple hours at a nice warm 37 degrees, E. coli B will grow. And so they won't grow lined up like this, like little E. coli soldiers. In fact, what they'll do is they'll grow into little piles, the beginning of colonies. You won't be able to see them yet because they just went from 1 to 2 to 4 cells, but they're the beginning of a colony. And so what I drew here was 4 little cells where that one was, and so 4 little cells, right? And they grew clonally. And so this one cell became these 4 cells and they're identical, right? And so that happened on both plates, whether the mutation is induced or spontaneous. And so the next thing that we did is we respread one of the plates, which means we left these 4 microcolonies alone, but here we took these 4 microcolonies, we broke them up, and we took these cells and we spread them all over the plate again. Just when they were getting comfortable, we decided to move them around. And then, right at the end of the day, at the end of the lab, we added T2. Now we did it with top agar, of course, instead of by spraying them, but whatever it is, you add the selective agent. And so here is where things can change. And so let's see what happens. If the mutation is caused by T2, if it is induced, then it is the addition of T2 that causes the mutation. And so now you will begin to see mutants over here. If the mutation rate is 1 in 4, then out of every 4 cells, you get a mutant. And so since these are piles of 4, then the odds of getting a mutant are 1 in 4. And so on average, then you'll see a mutant in each one of these piles. Now again, you know, you might see one in some and not at others by statistics, but we're really dealing with millions of cells, so it will average out. And we're just dealing with averages here, right? So out of these 4 cells, we'll get 1 mutant. And out of these 4 cells, we'll get 1 mutant. And out of these 4 cells, we'll get 1 mutant. And out of these 4 cells, we'll get 1 mutant. Remember, they're on top of each other. They're not lined up. We have what were originally 4 piles of 4 spread out here. And so we have 16 cells. How many mutants are we going to get? Well, 1/4 of them, so a total of 4 cells, but instead of lined up, they're going to be spread all over the place. Wherever they happen to land, we're going to get 4 mutants. And so T2 is going to kill anything that's wild type. The mutants are mutants because they are resistant to T2. And so when we incubate these overnight, what's going to happen? Well, the resistance continue to grow and divide, and the daughter cells are identical to the parental cells, right? The mother cells, and so they are resistant, and so you end up with piles of cells coming from these original cells, and what you see is that pile grows into a colony, and that cell becomes a colony, and this cell becomes a colony, and that cell becomes a colony, and so on, and these 4 cells become 4 colonies, and you get roughly equal number of colonies on both plates. They may not be exactly the same because of random deviation, but we had 4 here, we had 4 here, we have roughly 4 colonies on both. That's what happens if the mutation is induced. What happens if the mutation is spontaneous? Well, did the mutation occur at the same time? And so if the mutation is spontaneous, the mutation happened here after we added the T2. Well, the answer is no. The mutation could have happened any time prior to the addition of T2. Could have happened here or here or here, or well before we even put them on the plate, because time extends infinitely in front of the addition of T2. And so there's a lot more than 2 hours in front of the addition of T2. Let's presume that it happened at least 2 hours in advance. So what are the odds that of these 4 cells, one of them is a mutant? Well, if the mutation rate is 1 in 4, then of these 4 cells, one of them should be a mutant, because we were growing these cells up before we put them on the plate. And of the 4 cells on here, what are the odds that one of them is a mutant? Well, 1 in 4. So let's make that one a mutant. And then when we grow them up for 2 hours, all of the daughter cells of all of these are going to be identical, which means this mutant makes a mutant microcolony, the wild types make wild type microcolonies, and same here. And then we mess with them, right? One plate we leave alone, and the other plate we're going to go and we're going to spread all of these microcolonies around. The wild types are going to get spread around, the mutants are going to get spread around, and they're going to get intermingled. And so we add T2. T2 is going to kill anything that isn't a mutant. And so that means these guys are going to die, and these guys are going to die, and these guys are going to die, and all of these wild types are going to die. But 1, 2, 3, 4, these 4 cells aren't going to die. And this microcolony that came from that one cell, that isn't going to die. And so what are we going to see after incubating it overnight? Well, this one cell that's growing into a colony is going to continue growing into a colony. And so we'll get a colony there. This cell that grew into a colony that we broke up into 4 individual cells again will continue to grow and form 1, 2, 3, 4 colonies. And so what we'll see if the mutation is spontaneous, if it happened before this incubation and then addition of the T2 that we did after respreading it, what we will see is more colonies on the respread plate than on the non-respread plate. And so this is how you can tell. The key here is the moving of all of these microcolonies, breaking up of the microcolonies. not just that there's more or less, more or the same. You have to understand why. Make sure you explain that in the discussion. Make sure you understand it. It helps if you draw this out. Okay, so we know which plate to use now, right? We know that we want to use the respread plate, excuse me, the not respread plate, not the respread plate, because that was the actual number of original mutants. And we next want to think of, well, how much did we originally use? Well, if we want to use 50 as our number of mutants, we know how much we plated, 100 microliters, then the calculation is pretty simple. The mutant concentration is 50 mutants, or 50 mutant colony forming units per 100 microliters. 100 microliters is 0.1 mils. That's a nice way to think about it. It's going to make the math, the next step, easier. So 50 mutant colony forming units per a tenth of a mil, or 100 microliters. We say colony forming units instead of mutant cells, even though we were just talking about how each of those colonies come from a cell, because we don't strictly know that each one of those colonies came from a cell. What if two mutants land right next to each other on a plate? What are you going to get? Just one colony. So it's possible that two mutants landed next to each other. What if some cells are dead that should have been alive, right? And so you end up skewing numbers that way too. And so this is an allowance for the fact that there are things that can change the number of cells, in change between the number of actual cells to the actual number of colonies. Okay, so mutant colony forming units. So 50 per 0.1 mils. Now we want to normalize that to a full mil. So this is a tenth of a mil. How many tenths fit in a full? 10 of them, right? So that means 10 times 0.1 to make it a whole. 50 times 10 is 500. So what we have is 500 mutant colony forming units per mil. Now, we really, really want to use scientific notation in this lab, so that's 5 times 10 to the 2 mutant colony forming units per mil. It will save you a lot of trouble if you use scientific notation, not to mention points on lab reports and exams. But scientific notation will save you from having to use calculators and making mistakes. Trust me. You also want to make sure you include units. It's important to recognize units. This isn't elephants per ballroom or whatever. It's mutant colony forming units per mil. It'll make your life easier and help you keep track of things. All right, so that's one of those numbers. What's the other number? Well, the other number is going to be the viable cell count. So where does that come from? Well, we took our E. coli and we diluted it. How did we dilute it? Well, we took 10 microliters, put it in 990, and then mixed it, and then we took 10 microliters from this tube 1 and added it to 990 microliters of sterile media, and then we mixed it, and then we took 10 microliters from there and put it into tube 3, which had 990 microliters of sterile media. And so it started out very dense. It became less dense in tube 1, even less dense in tube 2, and much less dense in tube 3. And then the idea was to plate it. And if we plated some of each of these samples, what should we find? We should find bacteria coming from tube 2, and what? Even fewer bacteria coming from tube 3. How many fewer? Well, that's the question, right? How much is each one of these dilutions? Now, we didn't actually do this plating here from tube 2, but can you figure out what this dilution is? If so, you can figure out what the number of bacteria should be, the number of colonies should be different. That should be an easy problem. So this is what you actually did. And so what is each one of these dilutions? Can you go back and figure out that math? Well, each one of these dilutions is a 1 to 100 dilution. Here's how you figure out a dilution. You take what you're transferring, in this case 10 microliters, and you divide it by what you transferred it into plus what you transferred. So 10 microliters, that's what we're transferring. We're adding it to 990 microliters of diluent plus what we're transferring. So that's the total final volume. So the transfer volume plus the total final volume, over the total final volume. So that's 10 microliters over 990 plus 10. That's 10 microliters over 1,000 microliters. The microliters are going to cancel out. And so the final dilution, a zero cancels out. And so the final dilution here is 1 over 100. And so each one of these is a 1 to 100 dilution. That's 1 to 100, that's 1 to 100, and that's 1 to 100. Which means if you get a certain number of colonies from here, how many colonies would you expect to see when you plated from here? More or less? More, right? Because this should be fewer. And it should be how many fewer? It should be that many fewer. So what should you find here? 100 times more when you take a sample from it. Right. And how many would you find here? 100 times more. And so how many would you find here from here? 100 times 100 more. And so how many would you find here? 100 times 100 times 100 more. Or 10 to the 2, that's another way of saying 100, times 2, that's another way of saying 100, times 2. So 10 to the 2, 4, 6. 10 to the 6. That's the dilution. Okay, so how do we use this? There are some formula that always work. Okay, this will work on any dilution. This is the one that we just did. If each dilution is 1 to 100, then each step reduces it by 100, unless you're looking the other way, in which case it's increasing it, right? But this is what we did. So each concentration is 100 times more. That's what we were just talking about. A dilution is the volume of the stuff being diluted over that plus the volume of the diluent, the stuff that you were adding. That's what we just covered. But you need to be able to convert that to something called a dilution factor. That's going to allow you to use that in calculations to figure out serial dilutions. And so a dilution factor is just this flipped upside down. So a dilution factor is 1 over a dilution. So if your dilution is 1 over 100, or 1 to 100, then your dilution factor is 100. And so you're always going to be better off solving your dilutions so that you have a 1 over them. Always, always, always. Because you're going to want to get to the point where you can flip them upside down. This is a lot easier to solve to 100 than something you haven't, to flip to 100 than something you haven't solved. Okay? And then this. Start every problem that you have with writing out IC equals FC times DF. Initial concentration equals final concentration times dilution factor. In any dilution problem, you're going to know 2 of these and have to figure out the third. You can't do it if you don't know 2 of these. So you're always given 2 of these. Okay? There are problems that you can do. The dilution problems are in the Newcombe lab, and then there are videos that work them all out, and there are the written answers on Blackboard. Do the problems before you look at the solutions. And then if you still have trouble after working them out, come see us for help. Dilution problems are the kind of thing that seem impossible and then suddenly become easy. It's always like that. This is simple arithmetic. It's not calculus. You all have to pass calculus. They are kind of a weird word problem type of thing. It's not the math. It's the word problem difficulty, which means that when you get help, all of a sudden it'll become easy. So trust us, work on them, and then when you get stuck, come talk to us. We're going to be able to get you through it, and then there'll be free points on quizzes and exams for the rest of your time here in biology. All right, because you're all going to be able to do it. It's easier math than the other stuff you do in other classes, I promise. All right, so in this particular lab, how are we going to use it? Well, you have your dilution plates, what you diluted to 10 to the negative sixth, right? IC equals FC times DF. So you're going to need to count your colonies. Ideally, something between 30 and 300. That's what the lab manual says, right? Sometimes you don't have something between 30 and 300. That doesn't mean you throw out whatever you do have. You know, if all you have is 20 and 18, or all you have is 400 and 512, then that's what you got. You use the best you can, right? The 30 to 300 rule is when you have choices, use the one between 30 and 300. It's not throw things out if they don't fit these categories. So let's say, though, that you have 118 and 132 on your two plates. Well, you do it in duplicates for data reliability. They're both the same, so you're going to average them. So 118 plus 132 divided by 2 is 125. So you had an average of 125 when you plated on a 10 to the negative 6 plate. IC equals FC times DF. What do we need to find? I told you we're always going to know two of these. This is the initial concentration, final concentration, dilution factor. Well, let's see. We're going to get the final concentration by counting these. So we know that. We know we're going to be able to get that. The dilution factor is, well, we know what that is. That's from doing the dilution, which we did. So we know we're going to be able to get these. That means that's the one that we're going to need to find. All right. There's always two that we have. So excellent. All right. So you can start with FC or DF, whichever you prefer. Let's start with FC. So we know that we have 125 colonies. What was the volume? Well, let's see. Usually we plate 100 microliters. There are going to be exceptions. You have to pay attention to the exceptions. In this case, it wasn't an exception. We plated 100 microliters on both of these, and then we averaged them. So it was 125 microliters per, 125 colonies per 100 microliters. Okay. So this is just like the mutant side, where what we have is 125 colonies per 0.1 mils, or we can normalize that to a full mil, right? Because there's 10 of these in a full mil, which means multiply this by 10 and multiply this by 10. As long as you do to the bottom whatever you do to the top, or I should have said that the other way around, then we haven't actually changed this, right? So multiply by 10 over 10, and we get 1,250 colony forming units per mil. Same thing as the mutant colony forming units, right? We don't know for sure that a single colony came from a single cell, or that all of the cells were alive. So that's our fudge factor right there. And then we should turn this into scientific notation. 1.25 times 10 to the 1, 2, 3 colony forming units per mil. By the way, this is scientific notation, 1.25. I've seen some people do 12.5 times, right? So put the decimal point there, 10 to the 2. That's engineering notation. So it goes after the first number. Okay, so 1.25 times 10 to the 3 colony forming units per mil. That's our final concentration. So we got that one now. We need our dilution factor. So how do we get that? Well, the dilution factor is 1 over the dilution. We know we did 1 to 100 dilutions. So for each of our 1 to 100 dilution, the dilution factor is 1 over that. So 1 over 1 to 100 is 100. So 10 to the 2. The overall dilution factor is all of the dilutions multiplied together. So 10 to the 2 times 10 to the 2 times 10 to the 2. Remember, when you multiply numbers that have exponents, right, you add them together. So 10 to the 6th. So IC equals FC times DF. 1.25 times 10 to the 3 colony forming units per mil times 10 to the 6th. Again, you just add the exponents. So 1.25 times 10 to the 9th colony forming units per mil. There's our viable cell count. So now we have our mutation frequency and our viable cell count. Excuse me, our mutant concentration and our viable cell count. Now all we got to do is divide them, and we have our mutation frequency. So here we go. Mutant concentration, viable cell count, divide them, and we get our mutation frequency. So carrying it from the beginning, our mutant concentration was 5 times 10 to the 2 mutant colony forming units per mil. Viable cell count we just figured out, 1.25 times 10 to the 9 colony forming units per mil. Mutant frequency, we want to divide the small number by the big number, the number of mutants divided by the number of cells, or the concentration of mutants by the concentration of cells. If you get a number bigger than 1, it should tell you there's a problem, right? Because mutants are cells. Mils are going to cancel out. When you do the math, what you get is 4 times 10 to the negative 7th mutant colony forming units per colony forming unit, or mutant per cell. So this is a small number, right? Much less than 1. And that makes sense. Again, if it's a number bigger than 1, that would mean there were more mutants per cell, but mutants are cells. And so that should be the logic. Whenever you do a problem like this, you should think, does the number make sense? Well, there shouldn't be more mutants than cells if mutants are cells. So I hope this was helpful. I hope it makes it very easy to do the calculations. If you have any trouble, please come see a TA or come see me, and we will very happily help you out.